Hi everyone, I'm Charlotte Kilpatrick, reporting for VaccineNation from the World Vaccine Congress in Barcelona. Today I'm delighted that we're joined by Dr. Holger Heyn and Vijay Vaswani from Omniscope. Thank you for your time. To kick us off, would you kindly introduce yourselves and your current roles?
Sure. My name is Holger Heyn. I'm Chief Scientific Officer and co-founder of Omniscope. My background is really on the genomics side of it. And with Omniscope, we develop technology that reads very deeply into the immune system. And we use that insights to develop next generation diagnostics, but also inform therapeutic development.
Fantastic.
Yeah, thanks for having us. I'm Vijay, and together with Holger and Lynette, I'm one of the co-founders and the CEO. My journey has been in commercializing novel technologies in single-cell sequencing and sequencing, as well as diagnostics. But the one thing that we really wanted to understand was an important fundamental mechanism, which is the immune system. And particularly in the context today, how does our immune system respond to vaccination, to disease, which led us to building the largest database of the human immune system. And we're delighted to be able to put that to the service of vaccine developers today.
Fantastic, thank you. So you joined us in the immune profiling track to discuss AI-enabled immune profiling for accelerated immune development. Can you tell us a bit about how AI is contributing to vaccine innovation?
Sure, and obviously this is a very topical question. You know, the one of the most important things that we need to achieve in biology and in diagnostics and therapeutics is to be able to truly model our biology. So the way we went about it is to develop proprietary technologies to build a cell-by-cell database of the human immune system, spanning the breadth of our population and stratifying that by the way they respond to infection as well as vaccination. And obviously, with the innovation in vaccines and the importance of it as a public health initiative, there is a huge, huge amount of data out there. So what we've actually done is turn that biology, cell by cell, into a machine learning model. And this is what we actually launched at SF Tech Week, where we launched Immune GPT, our large language model of the human immune system. There, by applying interpretive AI, you can understand what your immune system is fighting or how it's responding, for instance, to a vaccine. But our team has also developed very sophisticated generative AI so that we can actually look at those cells, engineer and model them in silico so that we can use them as a therapeutic candidate or a cellular therapy. So those are some of the applications, but in particular, I think with vaccines, the ability to use these models to massively accelerate the process is key, particularly because that is a problem that hasn't been solved, but we may be able to contribute to solving that somewhat.
Fantastic, that's very exciting, thank you. Could you tell us about the main applications of your technology?
So by reading very deeply into the immune system, so we mainly look at the adaptive immune system, so T and B cells, and we read many of those. So we read like up to 1 million cells per sample and time point. And this really allows us to generate a high resolution picture, how the immune system is changing over time. For example, how it is responding to a vaccine. And here we contribute basically to the entire value chain of vaccine development from the very early stage, where immunogens are prioritized, to the very late stage, where we look at population surveillance, how vaccines worked in the population and potentially inform about booster shots. So here working at cellular level really allows us to have a quantitative tool, what kind of intensity of the immune system has been activated, but also to track it over time. So it's an early quantification that allows us to make vaccine design and prioritization quicker and the entire process more cost efficient. But then also in the later stage, into clinical phase on population surveillance, with a very sensitive tool to follow up and to inform about, in the end, the longevity of the induced immunity against the vaccine.
Great, thank you. So as the speed of vaccine innovation continues to increase and indeed AI's uses emerge, how receptive are vaccine developers to your technology and the use of AI more broadly?
Yeah, in short, very receptive.
Great.
And we found this Congress to be a great place to have these type of conversations. You see, at the end of the day, vaccination and vaccine development is a race to save lives. And if artificial intelligence can allow you to run that race quicker and save lives, of course, our colleagues developing these therapies are receptive. But I think one of the elements which underpins receptiveness is how broadly we apply AI. And we're not just applying AI, we are actually reading the biology, sample by sample. So this really means that we deeply understand their approach because we're mining data from their samples. And the questions where I see a lot of receptiveness are both very early in the process. You know, how immunogenic is an immunogen? Well, we can actually quantitate that. We can tell you how many cells respond to that immunogen. What is the breadth and depth of that response? How long does it last? So you can answer questions upstream in the discovery process. You can understand questions in the preclinical and clinical phase, such as understanding, well, my candidate, which I'm now investing significant time, budget, and effort in, is it inducing a response early on? Is it worth following up? As Holger mentioned, because of the elements such as boosters and regimens, being able to understand how a dose or a vector may change the efficacy of my vaccine is something which we can really address cell by cell. So we see a great receptiveness, and we also see a receptiveness in the sense that as a community we understand collaboration. Vaccine developers are fantastic at developing vaccines. Companies like Omniscope are fantastic at understanding the immune response and applying AI. So I see it more as science is we all, and as collectively we can use AI to accelerate the way we take therapies to the public.
Fantastic, thank you. So you've touched on the importance of collaboration there, and we're very glad you've joined us this week. Can you tell us what you've taken away from your time at the conference? What's been the best thing about it, or what did you achieve?
I think collaboration is a key word here. I think, first of all, we had an amazing time so far. The conference is really bringing together people from very different areas of expertise with complementary expertise, where we form together synergies to develop drugs quicker and better, right? So a trend I've been seeing is that in clinical trials and also interaction between, for example, a pharma developing a vaccine and a biotech developing a correlate of protection like us, there's much more kind of implementation of novel technologies like single cell technologies in clinical trials very early in the process. That will help speeding up the process. But also I think it's a very healthy interaction where one can form synergies to make a vaccine in the end better, the production more cost efficient. And in the end, it is what we always wanted to achieve, that we all, as Vijay mentioned, work together on a single goal and to kind of achieve that goal faster and better. Yes.
Fantastic. Well, I hope the rest of your time here is productive. Thank you for joining us.